Hey guys, I'm Shanti Das. Welcome back to my channel Mieosis. So recently I have updated the channel name from bioecstasy to Mieosis, and the reason is actually unknown. So that's not our focus. Um let's focus on our today's topic, and that is the extension of the GPCR pathway, and which is the cholera toxin pathway, and in the later video I will upload the pertussis toxin pathway. So today mainly I will discuss about the cholera toxin pathway. So cholera toxin is mainly acts through the GS pathway, which means it actually mediated through the CAMP pathway. So the cholera toxin is mainly a enterotoxin, which means that it targets the enterocytes of the small intestine. So it is known as the enterotoxin, and it is secreted by the gram-negative comma-shaped bacteria, which is known as the Vibrio cholerae. We all know that. And the cholera toxin is also abbreviated as CTX or CX or choleragen. So, and the receptor of the CTX is, or choleragen or cholera toxin is, gangliocytes of the enterocytes, which is the mainly the GM1. Okay, which is the GM1, which is present in the enterocytes of the small intestine. So let's come to the structure of this cholera toxin. So basically it is a AB toxin. That means the cholera toxin has two units, that is the A unit and the B unit, and under this it has the subdivisions like the subunits. So the A unit is actually divided as two subunits, A1 and A2, and the main catalytically active site is A1. Okay, this should be noted. And the B unit is actually subdivided into 5 subunits, that is the B1, B2, B3, B4 and B5. So it is also known as the AB5 toxin, right? So this is the structure of the CTX or the choleragen or cholera toxin. This is the A unit, and these all are the B unit. Okay, and under the A unit there is the A1 and A2 unit, and B1, B2, B3, B4 and B5 are the subdivisions of the B unit. So the A unit is actually catalytically active. This is actually catalytically active. So let's see how the cholera toxin is mediated by the GPCR pathway. Basically it is actually mediated by the GS pathway, which is known as the CAMP pathway. So basically this let's consider this is the enterocyte, right? This is the enterocyte. So this cholera toxin, which is released by the Vibrio cholerae, when infected, so it will bind to its receptor GM1, that is present in the surface or cell surface of the enterocytes. So after binding with the receptor, via the retrograde transportation, it will go to the endoplasmic reticulum, where it will be cleaved, and only the A1 subunit will be go further, because we said that the A1 is actually the catalytically active, and the other portions is actually helping in the binding with the receptor, nothing else. So the catalytically active A1 site will interact with the GS alpha GTP protein. So first we have to understand that in normal condition the G alpha GTP subunit is actually activated, or G protein is activated via the GS alpha pathway. Okay, so the adenylyl cyclase will be activated, and the CAMP pathway will be going on. So the CAMP will be activated, or CAMP will be produced, and the protein kinase A will be activated. So the protein kinase A will actually increase the CFTR. CFTR means the cystic fibrosis transmembrane conductance regulator, which actually regulates the different ion channels in the small intestine enterocytes. which is like the chlorine ion channel, the bicarbonate ion channel, water channel, or the potassium channel. These are the different types of ion channels which are regulated by the CFTR and via the GPCR or G alpha ACE pathway. So basically the A1 subunit actually does the ADP ribosylation in the G alpha GTP subunit. That means when the G protein is in activated mode, it is locked by the ADP ribosylation. Mainly ADP ribosylation is actually a post transitional modification. So the ADP ribosylation means the addition of the ADP ribose, okay, in this G alpha ACE GTP, or that means the activated G ACE protein. So it is the G protein is actually locked in the activated mode. So it will constitutively activate the adenylyl cyclase, the CAMP will be produced, the PK will be produced, and the CFTR is always active. So it will always open and the cation channels or the ion channels and the water channels will be always open. So the loss of different types of ions like chlorine, bicarbonate, water, and also the potassium can happen. So this will lead to the dehydration and it will lead to the cholera. So this is the proper mechanism by which the cholera toxin is accessed. So in this case we have some medications, and we have to give the oral rehydration solution, which is also known as the ORS, because of these dehydrated, because of the prevention of this dehydrated condition. So we have to rehydrate it. That's why we have to give the patient the ORS solution, and also the different type of medication which can be remove this Vibrio cholerae infection from the body, and the patient will be survive. So I hope you will understand the cholera toxin pathway by this video, and if you like this video, please like, share and subscribe. So we will meet in the next video. Bye.